Hello, my name is Annetta Linwood, and this is my genetic presentation on sickle cell disease. So what is sickle cell disease? It's an inherited red blood cell disorder that affects the hemoglobin. So what is the hemoglobin? It's a protein inside the blood that carries oxygen throughout the body. So when that mutation happens to the hemoglobin, instead of the hemoglobin being a circular shape inside of the red blood cell, now it becomes hard, sticky, and a sickle shape. And when it becomes a sickle shape, it affects the blood flow. That lack of blood flow creates a lack of oxygen to the tissues. So instead of the red blood cell being a circular shape, it becomes a sticky shape and gets stuck, which slows down the blood flow. Now, as I stated, that lack of oxygen to the tissue can sometimes create a sickle cell episode, which are episodes of pain. Now, there are different types of sickle cell anemia. There's HBSS, which is the most common and most severe. It's inherited when two sickle cell genes. That means you have one from each parent. That's called sickle cell anemia. You have HBSC. It's inherited one sickle gene from one parent and a gene of abnormal hemoglobin from another parent. You have HBS beta thalassemia, which is inherited one sickle cell gene from one parent and a beta thalassemia from another parent. Then you have rare forms of sickle cell as well. But then you also have the sickle cell trait, which is HBAS, where you inherited one gene from one parent because you just have the trait. You don't actually have the sickle cell disease itself. So what causes sickle cell disease? So sickle cell disease is caused by a mutation in the hemoglobin beta subunit. So hemoglobin, the larger hemoglobin, is comprised of two beta units and two alpha units. So when there's a mutation in the beta subunit, that's what creates the sickle cell. So each subunit is coded for a specific amino acid, and that amino acid makes up a protein. So the beta subunit is coded for GAG. Now, when that changes, that mutation or that change happen, then it's coded for GUG, which changes that amino acid, which will change how that protein is coded. And when that change happens, instead of being a circular shape, it turns into a sickle shape. So just to kind of bring it down to a simpler understanding. So say you have ham soup, which is the normal red blood cell. So ham is spelled HAM. Now say for instance you change that H to a Y. Now you have yam soup. That has changed the whole soup. That's the same as with the normal hemoglobin versus the mutated hemoglobin. You have changed it from a ham to a yam. Now you have a whole different protein. Now you have a whole different soup. So that's to kind of help you to understand how that change occurs and how it's so drastic to change it from a circular red blood cell to a sickle because of that one amino acid. So how is sickle cell inherited? So it's an autosomal recessive pattern, which means that you have to have two copies of an abnormal gene must be present. That means you get it from one from each parent. So if you draw a Punnett square, it will give you the probability of if the child will be affected, if the child will just be a carrier, or if the child will be normal. So let's take a look at the Punnett square. So if you have two parents, a mother and a father, both are carriers, which means they have a big A and a little a. So the mother and the father are both carriers. That means they have a 25% probability that the child will be affected, meaning the child will actually have sickle cell disease. Then there's a 50% probability that the child will just be a carrier or just have the trait like them. And then there's a 25% chance that the child will be normal. They won't have a gene, the trait won't be affected or anything. They'll just be normal. Now you have two parents that one is normal, which is the A, so they don't have the abnormal gene. But then you have a parent that is a carrier. They have the little a. So what is the probability? You have a 50% probability that the child will be a carrier, just have one gene, or 50% probability that the child will be normal, not a carrier of any genes. And lastly, you have the parent who is actually affected with the disease. So they have both of the abnormal genes. And then you have a parent that is normal. So they're unaffected. There is 100% probability that each child will have that one gene, which is that small a. So it's 100% probability that that father will pass on his one abnormal gene to every child. So there is 100% probability. So when we go to the next slide, it'll kind of give you what a real world situation may look like. So you have Tom and Sarah. They had 4 kids. Tom and Sarah are both carriers of the disease. So as we went over in the Punnett Square, there's a 25% chance that they will have a child that's affected. There's a 25% chance they have a child. There's a 25% chance the child could be normal. And then there's a 50% chance that the child could be a carrier. Well, in this case, they have 4 children. Amanda is a carrier. Their son, TJ, actually has sickle cell. Stacy is normal. And then Jordan has the trait. Now, Amanda married Michael. Now, there's a 50% chance that that child will have the trait or that child will be normal. In this case, they had 2 children. Ethan is normal, has the A. And then Olivia inherited that abnormal gene from her mother. So she's a carrier. Now, TJ married Amy. TJ is affected. He has the disease. And then his wife is a carrier. So then there is a 50% probability that they can have a child with the sickle cell or with the trait. And in this case, he got the abnormal gene from his dad and then the normal from his mom. So he is a carrier. Stacy married Maurice, so when they have children, there's a 100% probability that their children will be normal. So that just kind of gives you an example of what a real world situation could look like. So just to give a little bit more information, so the prevalence of sickle cell, it affects millions of people around the world, but it's more prevalent in people with sub-Saharan ancestry, people from the Caribbean, Central America, Greece, and Italy. In the United States alone, there are over 100,000 people with sickle cell disease, and it mostly affects the African American community. There's 1 in every 365 African American births results in sickle cell disease. 1 in every 13 African American births result with the trait. And then there's 1 in every 16,000 Hispanic births that result in sickle cell disease as well. Now, what are some of the physical effects and traits? Some of the physical traits and signs and symptoms appear in the babies at 5 months. They have anemia, they have episodes of pain, swelling in hands and feet, frequent infections, and the frequent infections come from damage to the spleen due to the lack of oxygen to the spleen, so you can get frequent infections. Unfortunately, there is no cure for sickle cell. You can only treat the symptoms, the signs and symptoms, such as the episodes of pain and the infections. There have been some new treatments regarding bone marrow transplants where they replace the abnormal stem cells with normal stem cells. And unfortunately, the life expectancy of people with sickle cell disease is shorter than the normal life expectancy of the general population due to the complications from the lack of oxygen. It can damage the organs. You can have a stroke. So that lack of oxygen really affects their body and how their organs are damaged. And lastly, there's genetic testing for sickle cell. So babies are actually tested 24 to 48 hours after birth as a part of the newborn screening program that was started in the 1960s. So every baby in the United States is actually screened for sickle cell disease. Also, if the parents are known carriers, they could also get genetic counseling. And once they get pregnant, they could have the amniotic fluid tested of the unborn child to see if that child has the disease or has the trait. Or if parents are known carriers and before they get pregnant, say they're doing IVF treatment, they could actually test the embryo with pre-implantation genetic testing. So there are different ways you can test. You can test it as a baby. You can do genetic counseling after you have your baby, or if you do IVF, you can actually test the embryo to see if the embryo has the disease or the trait. And that is my presentation. I hope I explained sickle cell disease well, and these are my references, and these are the references for the photos. Thank you.